Hello, welcome back. Today we'll be covering skeletal muscle. So the muscle cells are a mesodermal origin, and they make actin and myosin. The mesenchymal myoblasts will fuse, and they will form the myotubes, which will differentiate into the striated fibers. And then the muscle satellite cells are the reserve progenitor cells. Those work when the muscle has undergone trauma, and they work as stem cells to differentiate and replenish and heal the muscle. The myofibrils are made out of, they make up most of the cytoplasmic volume of the muscle cells. And hypertrophy is muscle growth by increasing in the diameter of the filaments, and hyperplasia is muscle growth by increasing the number of cells. So hypertrophy happens after the body has reached like adulthood, like there's no more development, right? So this is the only way that like we can bulk up and gain muscle, right? Hyperplasia is normal during childhood when you're still growing, but it becomes abnormal once you're an adult and you've finished growing. This would be indicative of cancer, especially cancer of the muscle cells. So the collagen in the connective tissue layers will transmit the mechanical force and help to keep the muscle cells like intact during contraction. The myoblasts divide and coalesce, forming the myotubes. I think I said that before. The myotubes are multinucleated and centrally located. They make the proteins that make up the myofilaments that gradually displace the nuclei to the periphery. Some of the muscle cells some of the myotubes don't fuse and differentiate, and instead they remain as the mesenchymal cells, which are the satellite cells. Those are located beneath the external lamina. They proliferate to regenerate muscle fibers after injury. Okay, so the perimysium is a thin connective tissue that surrounds each fascicle, and the fascicles are the functional unit of muscle. The epimysium is the external sheath of dense, irregular connective tissue, and it surrounds the entire muscles. And the septa go into the muscle itself, into the in between the fascicles, and they carry capillaries from the network, or they carry nerves and vessels, not just like capillaries. They can also carry like veins and arterioles. And then the endomysium that will surround like each individual fibers, and those have the capillaries that form the network inside, usually for like some kind of gas exchange, nutrient exchange. And all three of them are continuous with the tendon at the myotendinous junction. The mitochondria and the sarcoplasmic reticulum are found in between each myofibril, and the myosin is the thick filament, and it's made out of 2 heavy chains and 2 light chains, all twisted together with a globular head on the light chains. G-actin monomer also have a binding site for myosin, and the G-actin is part of actin, and the thin filaments, actin filaments, are made of tropomyosin and troponin. The muscle works for movement, joint stabilization, posture, heat generation. They also regulate organ volume. So your liver can only be a certain size, right? They move substances within the body via like peristalsis, And the four different types of movement is the contractility, how much the muscle is able to contract; extensibility, how much the muscle is able to extend; elasticity, which means how much, like, um, how able the muscles are able to bounce back after something like hyperextension or whatever; and then excitabilities, that means how how sensitive the muscle are to nerve impulses. And these are the different kinds of contraction. The isometric contraction, that's when you're just holding a weight. Concentric is moving and moving the weight towards the origin of the muscle, so the muscle will contract. Eccentric is moving away. Contraction, there's a sliding theory, pardon me, and that means that, like, the bands will slide over each other and contracting, and that is how the muscle shortens. The A band doesn't get any shorter, and it's dark because it contains both actin and myosin. The I band is the light band. It only has thin filaments. It doesn't overlap with thick filaments. It attaches to the Z disc via the alpha actinin or nebulin and titin proteins. Nebulin regulates the length of the thin filament, and titin connects the myosin to the Z disc, which keeps the filaments in alignment, and it works as a passive stretch resistance of the muscle fibers. So here's the sliding theory. Myosin will bind to actin at a certain site, making a cross bridge. After calcium has bound to actin and moved an inhibitory factor out of the way. So the power stroke is how the filaments slide and contract, and then ADP and an inorganic phosphate is released, and then ATP comes in, attaches to the myosin, and the myosin head was able to detach, and then ADP and an inorganic phosphate come in to, like, prepare the myosin for another stroke, and then calcium will come in, bind, move the inhibitory protein out of the way, and then it will start all over again. So every other band shortens as the Z lines are pulled together except for A. The M line will have the myosin binding proteins, and M lines are like straight down the middle, and they have the myosin binding protein myomesin that holds the thick fibers or filaments in place, along with creatine kinase. Remember when we were going through creatine kinase, that was an important enzyme in muscle because it breaks down like creatine, and that can have like a reserve of energy in the muscle cells. And ATP is required to release everything, of course. The sarcoplasmic reticulum contains the calcium pumps, and the sarcolemma is the cell membrane. The T-tubules are invaginations of the sarcolemma into the sarcoplasm, and it allows for the calcium to release and a uniform contraction of all the myofibrils. So remember how, like, there was all like connected into a big bunch? It wouldn't make any sense if only the outside can contract. So those invaginations allow the inner fibers to contract as well. I think I spelled this wrong, but in the skeletal muscle there's a triad, so there would be two terminal cilia along with the T-tubule, and that triad will depolarize the sarcoplasmic reticulum and release calcium. At the end of the axon, In the muscle is the motor end plate, and it's the neuromuscular junction, where the axon will synapse with the skeletal muscle fiber. The motor axon terminal contains a bunch of mitochondria, which enter the endomysium and lose the myelin sheath, forming the terminal buttons, right here. And the terminal buttons vesicles will contain acetylcoenzyme, or not acetylcoenzyme, I'm sorry, it will contain acetylcholine, and the acetylcholine will open the cation channels to depolarize, and then the cell will send the nerve impulse when, the Na or the sodium channels are open, and then acetylcholinesterase will remove the acetylcholine. So it will remove all of this, sequester it back, and then the cell is ready to go for another impulse. The individual fibers contract all or none in muscle, of course, and the fibers don't contract at some times to vary the force of contraction. So depending on how strong the contraction is, that just doesn't mean there's a halfway or a weak impulse. It just means that some of the fibers are not going through an impulse or a contraction right now. Hope that made sense. Okay, so calcium is released from the sarcoplasm. It diffuses into the myofibrils and bind to the troponin, causing the troponin-tropomyosin complex to move. So the troponin is the inhibitory factor on actin. So, tropomyosin is the inhibitory factor on actin, because calcium will bind to the troponin will displace the tropomyosin. That allows the myosin heads to bind, forming the cross bridge. The cross bridge will shorten the sarcomeres and generate tension. And then each cross bridge cycle occurs at different moments, so they're not all at the same time. So it allows the muscles to contract smoothly. And then the sarcoplasmic reticulum will actively reuptake calcium, and then contraction ends. Okay, so after death, calcium ions will leak out of the sarcoplasmic reticulum and allow the myosin heads to attach. But that doesn't cause the rigor mortis. Rigor mortis occurs about 24, 72 hours after death, and that's when the body is very, very stiff, and you're unable to move the joints. Important to know for forensic pathologists, that just kind of shows you when the patient has passed away, what position of the body they were in. And rigor mortis occurs when the cross bridges can't detach because there's no more ATP being produced. In the muscles, there's a lot of proprioception, of course. One of them is the Golgi tendon organs, and those detect changes in tension through the sarcomere. And it works to inhibit motor nerve activity if the tendon strain is excessive, and it regulates the amount of effort required. This protects the muscle from contracting too forcibly, causing some kind of avulsion of the bone or tears in the tendon, and of course the tendons don't heal. Myasthenia gravis, that is a neurodegenerative disorder where antibodies block the receptors in the sarcolemma, not allowing the acetylcholine to bind. And it's not really block the receptors. The antibodies are actively destroying these receptors, which affect the synaptic contraction. And the first sign is the ptosis of the eye. So it's the droopy eyelid. This person, while they may be awake, is barely able to keep their eyes open. Over time, there's going to be weakness in the arms and legs. There's going to be a change in the voice. And then, like, lastly, there would be, like, swallowing difficulty. And the patient loses a lot of weight. They're just too tired to even just chew. Dystrophin, it's a cytoplasmic protein that links the cytoskeleton to the sarcolemma, externa lamina, via this protein, the laminin protein, and the sarcolemma is stabilized by the dystrophin associated glycoprotein, or the DAG complex. It supports muscle fiber strength and transmits the force generated in the sarcomeres to the extracellular matrix. And it's involved in the Duchenne Muscular dystrophy. This particular disorder is X-linked, and dystrophin associated glycoprotein is affected, and that is because no dystrophin is made. And the progressive muscle weakness and degeneration occurs, and is really characterized by pseudo hypertrophy of the calves and the buttocks and the muscle by replacing the muscle with fat. So it looks like these people have been like working out quite a lot, but in reality it's not muscle, it's just adipose tissue. Other symptoms is like lordosis or scoliosis. Eventually there's going to be paralysis, and these patients do suffer some cardiomyopathy, and they have frequent falling, and they have a protruded belly because of the lordosis. They have a tiptoeing kind of walk because of the contracture of the Achilles tendon and the calf muscle being affected. And they are treated with some glucocorticoids such as prednisone or deflazacort. I'm not really familiar with that one, but I definitely know prednisone is one of the treatments for it. And it slows the disease progression if it's given early, like elementary age. It does improve the muscle strength and function. Physiotherapy does work as well to increase the muscle strength and to actually build the muscle versus replacing it with fat. Unfortunately, this is degenerative and there is no cure. Okay, so the different types of muscle fibers. Type 1 is a slow twitch fiber. This says that is associated with endurance, and not really. By endurance, it doesn't really mean something like walking. These are like muscle fibers found in muscles that are associated with posture. Those muscles are the ones that are like constantly working to keep our core upright. Type 2A are the fast twitch oxidative fibers, and they have a moderate fatigue rate, and they're used for moderate distances and intensities. And type 2B are the fast twitch or the glycolytic fibers. They're used in short distance and a high intensity, and they have a very high and quick fatigue rate. And this is a little like I guess misleading, because the muscles, like all the muscles in our body aren't just like divided by like type 1, type 2A, type 2B. Like all of these are mixed in with our muscle. It just depends which ones are recruited, like which ones are needed. So when you're scared, every single muscle fiber in the muscle is recruited, type 1, type 2A, and type 2B. That's of course to like produce the most amount of energy possible to either respond to the stimuli, whether you are going to be running away or standing to fight. So the more precise muscles will have more motor units and more innervation, and the motor units are all recruited from small to large, so it's all from like slow to fast twitch as contraction increases. Okay, so contraction is just the conversion of chemical energy into mechanical energy. Tone is the base tension, it's the low rate of nerve impulses to keep the muscles semi-active, ready to use. So my analogy is like putting your laptop to sleep, just closing the top. And then when you open it back up, you tap the keys a couple times and it turns right back on very quickly. And because there's like a low amount of energy running through, keeping it primed, so whenever you open your laptop, it's ready to go. This is in contrast to turning your computer off, like shutting it down completely. And then when you have to turn it back on, it takes forever. You have to wait for it to load, for everything to come back on, and then you have to type in your password, and then the computer is ready for you to use. So that's how the muscles work. It doesn't want to, shut everything down and turn everything back on. That's not useful because we are in motion almost all the time. Whether you're sitting in a chair, it doesn't mean like you're completely motionless. You're still breathing, right? And if you need to shift or scratch yourself, it doesn't take a couple minutes for you to move. It takes only like a second. So part 2 is the sensory specialization. In the muscle, there are the neuromuscular spindles, which are stretch detectors, and those respond when the extrafusal fibers are distended by increasing the rate of passage of afferent impulses in the IA fibers to the spinal cord and the brain stem. And these spindles contain interstitial lymph and intrafusal fibers. They're in 2 different types. The dynamic nuclear bags fibers, those are sensitive to the rate of change, along with the static nuclear fibers, which will sense the final change, not the rate of change. These neuromuscular spindles work with different types of sensory and intrafusal fibers to mediate flexes for posture, and they regulate the activity of the opposing muscle group. And they also work in regulating tone. The annulospiral or the primary sensory endings are non-myelinated ends of large myelinated fibers, which are like, the IA fibers. And they wrap around the nuclear bag and the chain fibers, like here. So look, they wrap around in a spiral, versus the flower spray or the secondary sensory endings. They're the small myelinated fibers. They're type 2 sensory fibers, and they innervate the nuclear chain fibers only. So here's the nuclear bag. See how it's like kind of a bag? It's spindle shaped. Here's the chain. The dynamic nuclear bag fibers are innervated by the type 1A fibers, while the static nuclear bag fibers and the nuclear chain fibers are innervated by type 2 fibers. They're the afferent fibers. And under resting condition, most afferent nerve impulses aren't consciously perceived. So that means we're not constantly thinking about tone. We're not thinking like, oh, my biceps always need to have a little bit of activity. The 1A fibers will synapse with the alpha motor neurons, which stimulate the extrafusal fibers in the agonist muscles for contraction, removal of the stretch stimulus from the intrafusal stretch receptors in the agonist muscles. And so we don't want it just means that like when you're contracting, there's no stretching, so the stretch stimulus is just removed, and the alpha motor neuron supplying the antagonist muscles are inhibited via the 1A inhibitory interneurons. Okay, so the extrapyramidal system is in the cortex of the brain. The sensitivity of the neuromuscular spindle is modulated by the extrapyramidal system of the motor cortex, and the cortical spinal tracts made by the extrapyramidal systems do not cross the medulla pyramids, they just kind of go straight down. And they synapse with the gamma motor neurons in the spinal cord, and they supply the contractile parts of the intrafusal fibers to maintain tension. The gamma motor neurons will adjust the sensitivity of the muscle spindles, so it all plays a role in maintaining tone. Okay, so the Golgi tendon organs are located in the tendons, and the musculo-tendinous junctions, they're made out of specializations of 1B afferent fibers, and they sense the changes in muscle tension and the rate of change. Contraction will stretch the GTO and stimulate the 1B afferent fibers, and those will send impulses to the anterior horn of the spinal cord. So we're carrying an excessive load. The 1B inhibitory interneurons will inhibit the alpha motor neurons, so the contraction is stopped. And it's called the Golgi tendon reflex. And it causes the muscle to relax and protect the muscle from using excessively heavy loads. Like I said before, it's to protect the muscle from contracting too hard and causing avulsion of the bones or tearing of the tendons and the ligaments. So next time at the gym and you're carrying too much weights and you accidentally drop it, don't be embarrassed, I suppose. It's just your Golgi tendon reflexes protecting your muscles. Okay, I know that was a bit long, but thank you for your attention and good luck studying.